Hello dear students. This figure shows us a typical eukaryotic cell with nucleus in it. The nucleus of a human cell contains 23 pairs of chromosomes, of which one chromosome is shown here. It contains genes on it. Now this chromosome is made up of supercoiled compacted DNA. This is the DNA strand, and on which bases A, T, G, C are arranged. Now let us see how the structures of nucleic acid are made. There are two types of nucleic acids, DNA and RNA. DNA is double stranded structure, as you can see here. RNA is a single helix, as you can see here. DNA is double helix. RNA is ribonucleic acid and DNA, on the other hand, is deoxyribonucleic acid. They are both made up of nucleotides. So nucleotides are the basic monomers of which DNA and RNA both are formed. So let us understand the structure of any typical nucleotide in detail. It has got three parts. First is nitrogen base, carbon nitrogen ring. They are nitrogen bases. A, T, G, and in case of RNA it is A, U, G, C. In case of DNA they are A, T, G, C. They also have a pentose sugar. In case of RNA it is ribose sugar, and in case of DNA it is deoxyribose sugar. It is called deoxyribose because on the second carbon here, you can see here, oxygen is removed from the ribose sugar, and therefore it is called 2-deoxyribose sugar actually. So DNA contains deoxyribose and RNA contains ribose, both of which are pentose sugar with 5 carbons. And finally they have got phosphate group as shown here. So all of these three components make a typical nucleotide structure. Now let us see types of nucleotides. Purines are A and G. Ag is silver. So you can see a tip is given, purine is pure silver, Ag. Now purines, as shown here, have got 4 nitrogen and 5 carbon arranged in a double ring structure. They have got double ring structure. They are of two types, adenine and guanine. They are shown here. What is the difference between A and G? Let us see that. In case of adenine, NH2 group is attached at carbon number 6 here, and it is called 6-aminopurine. In case of guanine, NH2 group is attached at carbon number 2, as you can see here, and it has got oxygen attached at carbon number 6, as you can see here in the parent structural ring of a purine, and guanine is 2-amino-6-oxypurine actually. Now let us talk about pyrimidines. Pyrimidine bases are derivatives of a pyrimidine, and they are of three types: cytosine, uracil, and thymine. Pyrimidine contain 2 nitrogen, 4 carbon arranged in a single ring structure, what we call parent structure of pyrimidine. Now Let us first talk about cytosine molecule. NH2 is arranged at carbon number 4 in a cytosine, and oxygen is arranged at carbon number 2 on cytosine. In case of thymine, oxygen is arranged at carbon number 2. Oxygen is also arranged at carbon number 4, and CH3 is arranged at carbon number 5. So actually speaking, chemically thymine is 2,4-deoxy 5-methyl pyrimidine. Now in case of uracil, which is present in RNA, oxygen is on carbon number 2 and carbon number 4. So uracil is actually T2,4-deoxy pyrimidine. So these are the details about slight differences among the 5 types of nucleotides which actually make nucleic acids, DNA and RNA. In case of DNA, 2 strands of DNA make this kind of ladle-like structure. Let us see how. A nucleic polymer has got backbone sugar to phosphate bond here shown. New bases are added to sugar of previous base and polymer grows in one direction. Nitrogen bases hang off the sugar phosphate backbone, as you can see here. So this is a single strand of a nucleic polymer in which nitrogen bases are attached to make a polymer. Now let us see more interesting part of it, pairing of nucleotides to make DNA. You will be able to see that there are hydrogen bonds between two DNA strands. This is one DNA and this is another DNA. There are 2 strands of DNA in the figure, and hydrogen bonds here are joining the 2 strands of DNA. These are the phosphate molecules showing outside of the helix. Here you have pentose sugar, deoxyribose sugar, and these are nitrogen bases. And according to Chargaff, as I have already explained earlier, T always binds with A always binds with T with 2 hydrogen bonds, and G always pairs with C, and C always pairs with G with 3 hydrogen bonds. And this is how 2 antiparallel strands of DNA, 2 antiparallel strands of polynucleotides are held together using hydrogen bonds between them. And this is how the ladder actually is formed. This shows the typical helical structure of a DNA molecule. It is called DNA double helix as suggested by Watson and Crick for the first time. Here hydrogen bonds between bases join 2 strands of DNA, these ribbon-like structures here. These ribbon-like structures here shows 2 strands of DNA which make one DNA double helix. Okay. There are hydrogen bonds between ATGC which are at the center of the helix, and phosphate backbone is at the outside of the structure. Now please understand how, why it is so. Because phosphates are charged molecules and they are water friendly, hydrophilic. So they are protruding outside, and hydrophobic nitrogen bases are concealed inside of the structure, because finally DNA has to be suspended inside the cytoplasm where you have 70 to 80% of water in the cytoplasm. So DNA hides its hydrophobic structures of nitrogen bases inside of the helix and hydrophilic part of DNA. The sugar phosphate backbone is located outside so that it can be easily suspended in aqueous cytoplasms. So this is how a typical DNA structure looks like, and there are measurements given. One helical pitch measures 3.4 nanometer. Two adjacent nitrogen bases are located at 0.34 nanometer distance, and this distance is 2.0 nanometer or 20 angstrom, whatever you call. So this is how DNA structure is formed by arrangement of various nitrogen bases which make the final structure. Thank you so much. These are some questions for you to solve. Thank you.